Good morning, y'all. How's everybody doing? Good? Okay, so the goal today is to quickly go back over some of the stuff that we talked about at the beginning of our discussion of enzymes, because the stuff at the beginning I feel like everybody got pretty well, but I just want to review it anyway. And then spend a lot of time thinking about the last most pages of the activity where I think that I started to lose everybody, okay, or at least most of y'all, okay, and kind of reinforce some of the things that I'm hoping you're able to think through, at least help you focus on what I want you to be able to think through, okay? So we moved into thinking about enzymes and how enzymes can serve as catalysts. And we talked about that there are really four main roles of enzymes, right? And the first one is that they are catalysts. And catalysts, by definition, speed up chemical reactions. And they do so by decreasing the activation energy, right? So hopefully everybody remembers that, especially since we just talked about it 2 days ago, right, that we can think about a chemical reaction in terms of the progress of the reaction. So on the x-axis is proceeding from reactants to products. On the y-axis is energy, and you can define the energy however you want. We're going to think about it as Gibbs free energy. So the idea is if a reaction is spontaneous, the reactant started at a certain level, and we end at a certain level for the products. And this difference is the Gibbs free energy, right? But the idea is that as the reaction proceeds, it has to go through this high energy intermediate. So we call this the transition state. And the energy difference between the reactants and the transition state is the activation energy. And kinetically, that's one of the most important things that define how fast a reaction can go. The rate constant is related to the size of this activation energy. The larger the activation energy, the slower the reaction is because the smaller the rate constant is, okay? So enzymes work by creating an environment that can stabilize the activation, the transition state, thereby minimizing the activation energy, increasing the rate constant, and therefore increasing the rate of the reaction, okay? So as we're kind of thinking about how these enzymes work and thinking about ways the active site can be set up to help do this, what you should be thinking about is what does the transition state look like? What does the highest energy part of the reaction look like, which the enzyme might be able to stabilize to help go faster, right? To decrease that activation energy. Okay, another thing we talked about it does is it dampens the requirements for harsh conditions. And there is a question on your activity that kind of tries to emphasize this, right? There's a question that talks about how, as an organic chemist, we might think about activating a nucleophile and creating a better leaving group for nucleophilic attack, right? So using a hydroxyl group as a nucleophile, we need to deprotonate it. That could happen really well under basic conditions, but to create a better leaving group from a phosphate, it might be easier to protonate an oxygen. That requires acidic conditions, right? Both of those will only happen under conditions that really aren't very amenable to a cell, right? Organisms evolve in buffers that are pH'd around 7-ish, right? In the 7.5 to down to like 6 range. So to have a lot of hydroxide requires really basic conditions to deprotonate a nucleophile. To protonate something to be a better leaving group requires really acidic conditions. And both of those conditions are extremes outside of what physiology has evolved to use. So the idea is that the active site of enzymes can create these microenvironments that will allow this chemistry to happen even though overall the environment in the cell doesn't support it. Okay? The third one is we confer a really high degree of chemical specificity. So this is the idea that we only want a certain reaction to happen, right? So for glycolysis, for example, we talked about how the first step in glycolysis is putting a phosphate onto carbon 6 of glucose. If we put the phosphate onto the wrong spot, remember every carbon except the first carbon of glucose has an alcohol group on it. So if we were to put that phosphate on a carbon 4 or carbon 3 or carbon 2, glycolysis doesn't proceed. So the idea is that the enzymes create this specificity and make sure that the phosphate will only go onto carbon 6 of glucose in the first step of glycolysis, so the rest of the steps can proceed. Okay? Likewise, the enzyme hexokinase will not recognize other sugars very well. It recognizes glucose, not isomers of glucose. So it won't recognize fructose very well. It recognizes glucose. Okay? And then the last one, it enables a really high degree of control. And this is important, we'll see really when we get to metabolic pathways. The idea that under some conditions, we want the enzyme to be active, under other conditions, we don't. So the enzymes have really good ways of basically increasing activity or decreasing activity based on external stimuli. Okay, so these are the things that enzymes are really good at doing and need to be good at doing. All right, so here's the idea that they're catalysts, right? They decrease the activation energy. We talked about this. They can create specificity, okay? And this is something that will come up later in the activity as well, right? So there are these enzymes called proteases that catalyze the hydrolysis of a peptide bond, right? So you have a long polymer of amino acids making a peptide. We know that all of them are linked together by peptide bonds. These enzymes will hydrolyze really any peptide bond, right? So anytime you have that amide linkage, in theory, a protease can hydrolyze that. But we want specificity. We want to know exactly where the enzyme's going to cut. So there are these specificity pockets that will dictate what point in a polypeptide chain will get hydrolyzed, okay? So trypsin, chymotrypsin, and elastase are all really, really, really similar proteins that catalyze identical reactions in an identical way. The only difference is what this specificity pocket looks for. Trypsin looks for long chains, long side chains that have a positive charge at the end. Chymotrypsin looks for big, bulky, hydrophobic side chains. And elastase looks for smaller, typically hydrophobic, but can be hydrophilic, right? And it's all about what kind of a pocket is presented near the active site. We talked about stereospecificity. We then talked about the 6 classes of enzymes, right? And the first question in your activity had you go through and look at a reaction and identify which class there are. Oxidoreductases are often really easy to spot because they involve one of 2 cofactors, NADH or FADH2. Anytime you see those cofactors, immediately think oxidoreductase because it is. Okay? Transferases are enzymes that catalyze the movement, the transfer of one functional group from one molecule to another molecule. This is different than isomerases, which takes a functional group from within a molecule and moves it some other place within that same molecule. Hydrolases and lyases are related. Hydrolases and lyases both break covalent bonds. One of them uses water. Hydrolases specifically add water across a bond, whereas lyases break bonds without using water. And then ligases are kind of the reverse of hydrolase and lyase. Those 2 enzymes, those 2 classes break molecules apart, break bonds. Ligases make bonds. Okay? So understanding these 6 classes and being able to quickly identify what class is a good thing to be able to do. We talked about some cofactors and briefly talked about the difference between a cofactor and a coenzyme. Coenzymes are organic molecules. Cofactors tend to be smaller ions. Okay? Quite frankly, I don't care if you know the difference. Just understand these are things that are not covalently linked to the enzyme. They interact with an enzyme, and the enzyme relies on them, but they are not actually part of the polypeptide chain. That's the important thing to recognize about cofactors and coenzymes. We then talked about 5 ways that enzymes can catalyze reactions. Okay? General acid-base catalysis. This is by far the most common. The idea with general acid-base catalysis is that within the active site of an enzyme, within the scaffold that the enzyme presents where the chemistry happens, there is very likely to be a general acid and or a general base. These are just side chains that can serve to donate a proton or accept a proton. Most chemistry that happens in the cell involves moving protons around to some degree, right? So enzymes are really good at this by having side chains, by having functional groups presented at perfect spots to abstract a proton, to remove a proton from something, or to donate a proton to something, okay? Nearly every enzyme uses general acid-base catalysis to some degree. Some enzymes, that's all they do. Some enzymes use it as part of more sophisticated processes. Covalent catalysis, this is when there's a covalent intermediate that forms between the enzyme and the substrate at some point during the reaction. Today, we'll look more closely at the serine protease mechanisms. In that mechanism, we're going to see that these enzymes use a serine as a nucleophile that create a covalent bond with a polypeptide chain, right? So the fact that there's a covalent bond between the enzyme and the substrate means that this is an example of covalent catalysis. Catalysis by proximity. This is really important if we're dealing with an enzyme that catalyzes a reaction that involves two or more substrates, right? The idea is that for this reaction to happen, if two things need to bind together, or if two things need to come together for transfer of a molecule, transfer of a functional group, then they have to get close together, right? And if they're just floating around the cell randomly, it's really unlikely they bump into each other, and even less unlikely they bump into each other like with the right orientation. So catalysis by proximity is the idea that enzymes bring the two or more substrates, close together, and they orient them properly such that the reactive regions are close together. Okay? Metal ion catalysis, this is the idea that metal ions can participate in reactions in lots of ways. If a metal ion helps the reaction in any way, this is an example of metal ion catalysis. And preferential transition state stabilization. This is the idea that we talked about here with our energy diagrams, that if the enzyme can stabilize that high energy transition state and decrease its activation energy, then that's an example of preferential transition state stabilization. So honestly, a lot of enzymes, many enzymes use all of these to some degree. Some of them don't. Some of them really only use one or two, but some of them use all of these different types of catalysis. These are all features, these are all ways that enzymes have evolved to be able to increase the rates of chemical reactions. Okay, and this, I think, is where I started to lose y'all. Okay, am I right? Okay, so with this slide and this slide, what I was really trying to just kind of point out, and what your book was trying to point out, is that giving a couple examples, like looking at a reaction that's catalyzed by an enzyme and thinking about some of the features that the enzyme presents that really help this reaction proceed properly, okay, proceed in the way that it needs to. So ribonuclease A is an enzyme that catalyzes the hydrolysis of the phosphodiester backbone of RNA, okay? So if it binds to any single-stranded RNA molecule, it breaks the backbone through hydrolysis. So what class of enzyme is this? Hydrolase, good, yep. Right, it uses water to hydrolyze a bond, hydrolase. So what I'm hoping that y'all can do is look at a diagram like this, right? So just look at the reaction, and as you look at the reaction, identify what's happening. Because if you can identify what's happening, then you can identify features that the enzyme has to have to help it happen, right? So in this reaction, we see a polymer of RNA, and we know it's RNA because there's a 2' hydroxyl group. We see that the identity of the base is generic, which means it's probably not important what the base is. Okay, so we start off with an RNA chain that has shown 3 units. We use water, and in this reaction, the phosphodiester backbone has broken at this point. Okay, we notice that the phosphate that is between unit 1 and unit 2 here is still attached to the 3' hydroxyl group of this RNA, right? And we now have hydroxide here on the 5' hydroxyl of this ribose. Okay, so looking closely at this, thinking about what features the enzyme would need to present to be able to help this reaction proceed. The first thing we should think about is what features does the enzyme need to let the substrates bind? Okay, so in this case, the substrate is an RNA chain. Okay, and the idea here is this enzyme is not specific. It just looks for RNA. It looks for single strands of ribonucleic acids. And as long as there's water around and it interacts with this, it will catalyze this hydrolysis. So the things we're thinking about is that we need the enzyme to be able to bind to an RNA chain, a single-stranded RNA chain. And the place that interacts is probably not important because we are not identifying specific sequences here, right? We're not saying this is an adenosine, this is a guanine, this is a uracil. It's just a base, so it's generic. So the idea here is that the enzyme is going to be looking for features of an RNA chain that every sequence has, no matter what that sequence is. And what's the one unifying thing of all polynucleic acids? The charge on the backbone, right? The negative charge on the phosphodiester backbone. No matter what the sequence is of bases, there's always a backbone that contains a ribose, a phosphate, another ribose, another phosphate, another ribose, another phosphate, and these phosphates all present a negative charge. So the backbone is a polyanion with very consistently spaced negative charges. So I have a negative charge positioned right here. So at some distance away, there's another one. At the same distance away, there's another one. At the same distance away, there'd be another one. Right? So no matter what the sequence of RNA is, the idea here is that they all have a negative charge, multiple negative charges spaced at identical distances apart. So if we're trying to think about how this enzyme might bind to this sequence non-specifically, we're thinking that it's going to bind to that, like those negative charges, right? Not the bases, but the negative charges. This also makes sense because it's the backbone that the chemistry happens on, right? So when we're thinking about how this RNA molecule is going to bind, we're thinking about having an enzyme that presents a scaffold, a structure that perhaps has some sort of a groove where an RNA chain can fit in. And in that groove, there are positive charges spaced at the same distance that the negative charges are spaced on our RNA polymer. Right? And how that happens, how the enzyme does that, y'all don't need to know. Like that's not the point of this. What I want you to understand and to be able to predict is ways that the enzyme can do this. Right? The enzyme can recognize an RNA chain by presenting positive charges at distances, at spacings that are identical to the negative charge spacing on the phosphodiester backbone. That's how we can recognize non-specific RNA sequences. If we were trying to recognize specific sequences, then it'd be all about recognizing the hydrogen bonding pattern of the bases. Does this make sense? Any questions so far? Okay. And so then if you look at the enzyme, indeed you see a groove here, a channel where the RNA binds. And that channel goes right through, or it includes the active site, which in this case involves two histidines. These histidines serve as a general acid and a general base. And I'm not even going to talk about the mechanism here because it's not that important at this point. It contains a general acid and a general base. So the enzyme binds to RNA because it's able to recognize the negative charges on the backbone, and it places one of these phosphodiester linkages between these two histidines so it can be hydrolyzed. Okay? Another cool feature of enzymes is that oftentimes they can catalyze multiple reactions. Okay, so this enzyme tryptophan synthase actually catalyzes two distinct reactions, and it does so at two distinct active sites. Okay, so here in this first step, you take this molecule indole 3-glycerol phosphate, which looking at it, you can see that it kind of resembles tryptophan, but instead of having a polypeptide, or instead of having a amino acid backbone, it has glycerol. So in the first step, glycerol is removed and we're left just with indole. That happens at the active site here on the left with that, is that purple? That purple at alpha subunit. We produce indole, which then can react with the amino acid serine. Serine gets added on here, and that's how we make tryptophan. Okay, what we're just trying to point out here is that sometimes complicated reactions can happen in a single enzyme. And this one can actually catalyze these two steps. And it does so by preventing the release of indole. So once indole is produced in the alpha subunit, there's this tunnel that it diffuses across over to the beta subunit where serine is added to it. Right? This is a really cool strategy that's evolved this enzyme because it prevents the release of an intermediate which would then slow down the reaction, right? If the intermediate were released, the reaction would be a lot slower. So this can speed up the reaction by preventing the release of an intermediate. All right, that brings us to poly-A polymerase, which really is the focus of a big chunk of your activity, okay? So I think it's on page 3 of your activity. It kind of talks about, it presents the reaction that poly-A polymerase catalyzes, right? So the idea here is that the poly-A polymerase catalyzes the reaction where it takes a non-specific sequence of mRNA and it adds adenosine to the 3' end. Okay, and this is really important for processing mRNA in eukaryotes, right? In biology, you probably learned that all mRNA has a poly-A tail for eukaryotes. This enzyme is what does it. It finds the mRNA and it just starts adding on adenosine, okay? So what we need to have available to us is a non-specific interaction between the enzyme and the mRNA chain. As we just talked about with ribonuclease A, what feature of an mRNA chain is consistent but not sequence specific? Yeah, like the phosphodiester backbone, right? The spacing of negative charges on the phosphodiester backbone. So if the enzyme can recognize that charge, the spacing of those charges, then it can recognize single-stranded RNA chains. Okay, that's one of the substrates, the mRNA, the single-stranded RNA chain. The other substrate is ATP, right? It's on your paper. The other substrate is ATP. So this one is more specific, right? So it has to take the 3' hydroxyl of the mRNA chain, right? The very 3' end of it, activate that to be a nucleophile, get the nucleophile to specifically attack the alpha phosphorus of ATP. Not DATP, right? Not deoxy ATP, not CTP, not UTP, not GTP, specifically ATP. So this enzyme has to have the ability to recognize two things: a single-stranded chain of RNA, placing the 3' hydroxyl at the active site, and it also has to have the ability to recognize ATP and select specifically ATP instead of any of the other nucleic acids and instead of any of the deoxynucleic acids. Okay? So thinking about how that happens is important. And your activity kind of walks you through this on that first page where we look at it, right? So it asks you to think about what is the nucleophile, what's the electrophile, and the only way to do that is to look at the reaction. When you look at that reaction, here, I'll pull it up. Okay, so when we look at the reaction here, right, the important thing to be able to do is to identify what is happening, right? So in this case, we see that we have this RNA chain. We know it's RNA again because of the 2' hydroxyl. We're adding on ATP, and what we notice is pyrophosphate diphosphate leaves, and we've made a new bond between the 3' hydroxyl of the RNA chain, right? This 3' hydroxyl, and the first phosphorus, the alpha phosphate of ATP. So the bond is formed between this alcohol group and this phosphorus, which immediately tells you what the enzyme has to be able to do. It has to be able to orient these substrates such that these two atoms are close together, right? If it doesn't orient them so they're close together, the chemistry can't happen. Okay, so I think one of the questions a little bit further down says, what does the enzyme have to do? Well, it has to be able to orient these substrates. It also has to be able to recognize an RNA chain. It also has to be able to recognize ATP and specifically put it in place instead of the other nucleic acids. Okay, we've already talked about how recognizing a single-stranded chain of RNA can happen, right? The spacing of those negative charges. What do you think might help the enzyme recognize ATP instead of the other nucleic acids? So further down the activity, Okay, so hopefully, hopefully everybody can pretty quickly tell me how it can specifically recognize RNA, right? So ATP instead of deoxy ATP, right? What's the difference between ATP and deoxy ATP? Hmm? Yeah, the 2' hydroxyl, right? So to be able to select for ATP instead of deoxy ATP, it's probably looking for this hydroxyl group. So if you were asked to propose how the enzyme might do it, you could say, well, I know that on ATP, there's this hydroxyl group, and there's not that hydroxyl group on deoxy ATP. So the enzyme must have a hydrogen bonding partner with this hydroxyl group right here that will increase the affinity, increase how tightly the substrate binds when ATP is bound there. And if deoxy ATP happens again in, the affinity is a lot lower, so the chemistry won't happen, right? So the idea is that it won't bind as tightly if we can't make a hydrogen bond with that hydroxyl group. Make sense? Okay. So that's how we can select for ATP instead of deoxy ATP. Selecting for ATP over the other nucleic acids, over GTP, UTP, or CTP might be a little more difficult, right? Because especially selecting for ATP over GTP, like they're both purines. They both have the same ring structure. The only difference is what's present on the Watson and Crick face here, right? So for ATP, we know we have a hydrogen bond donor and a hydrogen bond acceptor at these two positions. Whereas with guanine, we have an acceptor, a donor, and a donor. So anytime an enzyme is trying to select for one nucleic acid instead of another, that's what you should be thinking about. You should be thinking about the enzyme being able to recognize the hydrogen bonding pattern on the Watson and Crick face, where we make Watson and Crick base pairs in DNA structure. That's the difference between the nucleic acids. Nothing else. At least for the two purines or the two pyrimidines. Okay? And then the difference between a purine and a pyrimidine really is the size of the ring structure, right? So our purines, adenosine and guanine, we know have two rings. It's a bigger ring structure, and our pyrimidines have one ring. So here it's really gonna be all about the size and spacing. So if we're trying to select for a purine, which has a two-ring structure, and not a pyrimidine, it's gonna be about the way that this binding pocket is shaped, right? Imagine like just a circle going around these things, right? So around our pyrimidine or around our purine. And based on the shape of that pocket, it can exclude either purines or pyrimidines. Okay, and you may remember from Monday, you may not, but you may remember that in the active site of this enzyme of poly-A polymerase, there's a magnesium that's positioned right here, right? It's positioned right here, close to that nitrogen. That nitrogen is probably actually making an interaction with that magnesium ion. And with that magnesium there, it prevents this six-membered ring from binding, right? Because this is where ribose attaches. And if there were a six-membered ring right here, it would take up more space at this position, and it wouldn't be able to fit because that magnesium's there. So the enzyme is able to select for a purine instead of a pyrimidine because of the placement of another structured piece of the enzyme, in this case, a magnesium ion. It could be anything though, right? You could propose that there's a methyl group from an alanine sitting here that prevents our pyrimidines from binding, right? The idea is it's about the shape of the pocket. The shape of the pocket will only recognize a purine and not a pyrimidine. The opposite would be true if we're looking to bind to CTP. If we're trying to bind to CTP, then we're going to have something in place where we have a big structured part of the enzyme right here, and that would prevent this larger ring from binding. It's about the shape, right? We are trying to recognize the shape of this ring structure. Okay, so now we've now set it up so we can only have DATP or ATP or GTP binding. How do we select between ATP and GTP? How does DNA do it? Why do we only get specific base pairs? Why does A only base pair with T and not with C? The Watson and Crick, right? So like the hydrogen bonding pattern on the Watson and Crick side of our nucleic acid base. So we know adenosine has a hydrogen bond donor here and a hydrogen bond acceptor here, and guanine has an acceptor, a donor, and a donor. It's a different pattern of intermolecular forces, a different pattern of hydrogen bonding. An enzyme can recognize that pattern, right? So if the enzyme were to present a hydrogen bond donor here, a hydrogen bond acceptor here, that would partner perfectly with adenosine and not with guanine. So if guanine tried to fit in there, it wouldn't be able to make the proper interactions and it would have a much, much lower affinity. Does it make sense? So at this point, I certainly don't expect y'all to be able to go look at an active site and tell me what's going to bind. All right, that's really hard. Oftentimes, experienced biochemists can't do it. But what you should be able to do, just using chemical and biochemical intuition, is to look at a reaction, look at substrates, and predict what features might be there that could help it recognize one thing instead of another. What features should the enzyme present that will help it to recognize specifically the 2 substrates or 3 substrates or 4 or whatever it is that need to be there? And then in addition, what features need to be in the active site to help the chemistry happen, right? So for ribonuclease A, we need to be able to activate the 3' hydroxy, for poly A polymerase, we need to be able to activate our 3' hydroxyl group to be nucleophile so that the reaction can start. So perhaps you can propose there's a general base in the active site that activates that 3' hydroxyl, that deprotonates it, making it an oxyanion, making it a better nucleophile. Yeah? All right. So with that, we'll go ahead and stop. I want you to work through, if you haven't already done so, work through this ribonucleic, the poly A polymerase part of the activity. And in about 15 minutes, we'll stop and we'll talk about serine proteases.